Good morning. Thank you all for coming bright and early to the first talk. So today I'm going to be going over some of the work that we have that's going on right now on the station. We have a collaboration between NASA and the European Space Agency to use a furnace called ISS EML. EML stands for electromagnetic levitation. Hence the title of my talk. This has been a 10-year ongoing thing, and we're about halfway through the scheduled number of runs that you'd expect to have over the life of this furnace. The work I'm going to be talking about today was from the first batch of samples that went up. We're currently finishing the second batch of samples, and the third batch of samples is ready to be launched and got up there to the station, scheduled for 6 total batches. So today's talk, I'm going to start off with a little bit about the motivation for this research. Then I'll go into some of the physical phenomena that are involved, talk a little bit about our modeling activities. I'm going to bring the results to a dimensionless presentation form, which allows us to really understand not only the behavior of a single alloy system, but then bring in other alloy systems as well so that we can compare the robustness of our models. And then finally draw some conclusions. What you see on the right there is a ternary phase diagram. Iron is at the bottom left. Pure iron is at the bottom of the left, and then you have mixtures of nickel and chromium that would therefore create different potential alloys. The alloy that I'm going to be talking about today is that batch 1 alloy that has the yellow star. It's down there right on the edge of the purple circle. That purple circle represents the family of alloys that are used in austenitic casting operations on Earth. The 2 green alloys are upcoming flight. These are the ones that we have on station and are ready to start processing, and they should be somewhere in the March to June timeframe. Those are at about, they're located along the 70 weight percent isopleth. That's where commercial stainless steels lie. So alloy 347, 316, 303, the workhorse stainless steels are of that composition. And then finally, we're proposing to do a sample that's off of those concentrations so that we really get an figure out the influence of iron content on properties and behavior of these materials. So what is the role of new materials in human exploration? Well, first off, need for in-flight and colonization, outpost maintenance, repair operations. So this activity would be additive manufacturing, which I'll be talking a little about later, and welding operations, and supports the reliability of the mission. Additionally, the new technology, we could have an enabling technology for developing new rockets and new combustion systems, and therefore this work supports commercial and aviation. And finally, next generation metal processes. How do we do mathematical modeling in order to improve the fidelity and the robustness of our casts and increase reliability while reducing costs? So this supports development of transformative technology here on Earth. So overall, this is why we look at alloy investigations. We want to optimize how we make materials. We want to understand the influence of convection. How does stirring influence how the defect structures form and how the metal solidifies, and be able to predict, based on changing conditions during processing, what the effect will be. Therefore, we're able to control our process and increase reliability. And finally, why does NASA care about solidification? Well, we want to make new hardware. We want to come up with new space technologies, especially in the additive manufacturing area. On the bottom, I included a couple of mathematical models just so that you see what the current capability is. That right now we can pretty much predict, this is a conventional mold filling operation where you're going to pour metal into a mold and the gas gets expelled through the mold, and so we can model where the gas gets trapped, so where you get porosity. We can also figure out what the velocity is during fills, so how much convection is there on a local basis and how that would influence defect structures. Right now, we understand how to model heat and mass transfer, but we really don't understand what the influence will be on transformations in the steel. And I'll be explaining, that's the main emphasis of my talk. And then lastly, we can model, well, where does the melt solidify last? Because that's where all the defects are going to be. You can see those two posts, the front and the back are there. Those are called risers. Those are there specifically to take the last little bit of fluid and hopefully gather in all the defects in an area that you're just going to cut off and throw away. So how do you design the mold is really important in order to get the properties you want. So what we do is I look at deep undercoolings and rapid solidification. And undercooling is bringing a liquid to below its equilibrium solidification point. So you end up getting a liquid that's cooler than it should be. It's a metastable phase. And that means there's a large driving force for solidification to some solid phase. And in the case of the steels, you can see if I were to take a liquid of the composition, say of 15 weight% chromium, and undercool it down there to where the blue dot is, the first thing that will happen is the temperature will rise up to the red dot. And you can see that on the right, on the far right, where the temperature has cooled down and then the temperature rises to a metastable melt plateau. That's the ferrite, a BCC phase. Then there's a delay time where the solid phase is still in equilibrium with the remaining liquid. And then finally, the second phase forms, and that's an austenitic phase, and that's your final phase. So we have a two-step process. So solidification occurs from the melt to the ferrite phase to the austenite phase. And the key thing is, as that phase is sitting on that melt plateau, things are happening in the microstructure. It's coarsening, defects are forming, and it's very important to understand what is going on during that period, and that will change the final properties of the part that you make. So what we'd like to have is a theory that can explain, well, how long is that phase going to be in its metastable form before the final transformation occurs? Now, I put a star there on the second transformation, and the reason I do that is that you'll notice if the blue dot had been, say, slightly higher up, it would still have the same second temperature rise. So you would have, instead of being deeply undercooled, you would be shallow undercoolings, as long as it's below the red dot. And therefore, you would expect that the transformation that would occur from the metastable phase to the stable phase always would have the same driving force, because the temperature is always the same between the red and the yellow dot, no matter how deep or how shallow the blue dot went. Well, I don't want to bore you with the math, but the bottom line is there is a driving force that can be calculated from the equilibrium phase diagram, and I put a star on that one. That would be the driving force that drives that second transformation. The key thing to take from this slide is the thing at the bottom, that the delay time, tau, is inversely proportional to this delta G sub V to the -4th. Delta G sub V is the free energy that drives this transformation. So if I were to double delta G, double the driving force, the delay time would go to 1/16 of its value. So I have a numerical prediction of the influence of how the driving force relates to the delay time. So remember that -4. Problem is, that's not what happens. We would have expected that no matter what the undercooling was, we would have a certain delay time indicated by that star. But it turns out if we change the undercoolings, as indicated by the red dots and the red line, then the delay time gets shorter and shorter and shorter. And that's not according to theory. Those red lines correspond to tests we do on ground using an electrostatic levitator at NASA Marshall. And there's no stirring at all. It's completely quiescent. When we do ground-based tests using electromagnetic levitation, there's lots of stirring. We can't stop the stirring. And those are the blue dots at the bottom. So you can see that not only does undercooling change the delay, but also stirring changes the delay time, and significantly. And no theory up till our work started could explain that. The open dots there, or the open triangles, are work at NASA Marshall using the electrostatic levitator, but shining a laser beam on the side of the sample, and that creates very, very small convection due to Marangoni effects. The surface tension will drive some stirring within the sample, but it's only a small amount of sample stirring that's possible. So what our desire was we wanted to go beyond our ground-based limitations. On ground, we could either get no stirring, a teeny little bit of stirring, or a ton of stirring. Well, it turns out that the reason we get a ton of stirring on ground is because the weight of the sample pulls it down into the coils. And you can see a picture there on the left. There's a sample that's contained within a cage so that it doesn't float off and not be processed. And then that cage is inserted between two coils. And those coils contain, they're copper, they contain water that's flowing to cool them. And we put a real high frequency current in there. That creates a magnetic field. That magnetic field then inductively heats the sample, so we can heat the sample. It also induces a lot of stirring, and on ground, gravity pulls the sample down into the coils and creates a lot of stirring. In space, there's no gravity. And so we can have the sample floating up there, and we actually don't have as much stirring. We can get a very, very limited amount of stirring, or we can turn the power up and get lots of stirring. So in space, we can investigate the full range of convection in between what we can do on ground, which is a huge advantage to us. So the approach is to share samples. We take some samples that, like I would do work on rapid solidification, another investigator would do work on thermophysical properties, and someone else would be looking at resistivity, and someone else. So we have one sample has multiple uses. Biggest collaboration we have is with University of Massachusetts. Professor Robert Hires helps us out by looking at what convection is within our sample so that we know when we set a certain power level to the coils, what the convection is in the sample. And so what you see here is we get recirculating loops, but the key is we need to know the density, we need to know the viscosity, we need to know the resistivity. So we need to measure these thermophysical properties. So here's a video of what we get was on orbit. I apologize for the flashes. The flashes were individual images that were dropped during transfer from space to ground, so there's nothing we can do about that. Those images were lost. You can see the sample is going to start to wildly oscillate. The blue curve is the temperature. We're now at the very high peak of that, and so there's lots and lots of convection in that sample. But now we're going to turn the power off. That's the red curve. We're going to turn the power off, and the temperature is going to start dropping. And therefore, the convection is going to start to decay down, and we're going to have a quiescent sample. Then you can see we're going to apply 2 pulses to the sample. That's going to squeeze the sample and cause it to deform. And I'll tell you when that happens in the video. You see it's quiescent now. And there we applied a pulse. And so we're looking at the top of the sample, and it's doing this. Okay, because we squeezed it, and now it's relaxing. And now we're going to hit it with a second pulse at a different temperature. There it goes again. And the frequency that oscillation occurs relates to the surface tension, and the time that it takes for those oscillations to dampen out relates to viscosity. Now you're going to see a flash. That corresponds to that rapid temperature rise. There it goes, right there. Now the sample is solid. So we do investigations initially on the thermophysical properties as a function of temperature. And then we do investigations on the rapid solidification event that occurred. Now, this video was taken here at about 150 hertz so that we could look at the movement of the surface of the sample. The next videos I'm going to be showing you are going to be at 40,000 frames per second. So you have to go really fast in order to see the solidification event. But first off, I wanted to explain what did we end up getting? Well, here you see on the left, we changed the control voltage, and that changes the amount of heat that we're putting back into the sample and changes the convection of the sample. So the green dots represent the lowest conditions we could get. We had it as quiescent as we possibly could. And on the diagram on the left, you can see as we turned up the heater current, that increases stirring. And the yellow represents transitional flow, and the red represents fully turbulent flow. When we plot the delay times as a function of those conditions, we see, as expected, when the convection is low, it approaches the Marangoni conditions that we saw on ground. And when convection is high, it approaches the convection that we see in the electromagnetic levitator on ground as well. So we're able to look at that whole range of convective conditions between quiescent to fully turbulent. So this video will show you the 30,000 frame per second exposures, and you can see growth of the first phase down there in the bottom of the image. And the BCC phase is growing across the liquid, and now you have a mixture of liquid and BCC phase. And there's a very long delay time. You can see where the green circle is up there at the top. It's a very long delay time before, there's the second phase right now growing across the first phase. So the images at the top show you, well, we had the first phase, then there was a long delay, and finally then we had growth of the second phase. That represent quiescent conditions, long delay times. Next, I'll show you one that's fast. So there's the first phase, and there's the second phase, almost immediately after it. So that's shown with the red circle. It's a much shorter delay time. It has both higher undercooling and higher convection. So now what we'd like to do is figure out a numerical method to predict this new influence of convection and undercooling on delays. Well, the analogy I'm going to use is very widely understood means of strengthening materials. It's called hot working, and you deform a material and introduce defects into it, and therefore it becomes less ductile but much stronger. If you do it while the sample if you warm that sample up, recrystallization can occur. And that's what the curve on the far right will show you, that you've taken this sample that has a lot of defects in it, you heat it up, and the strength will start to drop while ductility goes up as you form new crystals. So this phase transformation is induced by adding damage to the material. And in fact, the more damage you put in, the faster that transformation. That's exactly what we were seeing. So mathematically, we had this second transformation, which is indicated by the black bracket. And it's the same no matter how deep the undercooling was. If the undercooling is shallow, like the red curve, there'll be a small contribution to free energy. And if the undercooling is deep, like the blue one, there'll be a large contribution to undercooling. So this damage component is going to be linearly dependent on the undercooling. Don't yet have a numerical value for the effect of convection, but let's take one step at a time. Let's first look at just the effect of undercooling. Again, the reference condition is that quiescent condition with no stirring and no additional undercooling. So again, you go through the math. The key to this slide is at the bottom. The approach we use is to look at only the electrostatic levitation data, because that had no stirring at all. I don't have to consider that particular contribution to the transformation. When I look at that data, I can say, okay, I know how much free energy I would need in order to get that delay time if the transformation followed that driving force to the -4 rule I showed you. So I know how much free energy I need, and I can compare that to how much free energy I have available due to the undercooling, the initial undercooling of the sample. And when I do that and I plot them, the curve on the bottom is how much free energy did I have available. On the left-hand side is plotted, and how much did I need in order to see that delay time. And you can see it's a slope of 1. I need every bit of the energy that's available in order to cause the transformation to follow the observed experimental data. What this says is that free energy is retained, which conservation of free energy is something that we're all pretty well familiar with and could understand as being possible. Now, I put on the left that we did a bunch of tests for the bunch of different alloys. So the 2020 alloy is 60% iron and 20% chromium, 20 nickel. And then there's a 72% iron, 11 chromium, 17 nickel, etc. And this was true for almost all of the alloys. The 1216 is a little bit off, but not that you can see there was a lot of data scattered there. So the family itself shows that free energy is conserved when considering only undercooling. But what about convection? Well, now I look at the results in space and on ground, and that's what's plotted in the graph. You can see as a function of the amount of stirring that I did, the melt shear, how much damage-free energy did I need in order to see the transformation. And you can see that if I'd only had the brown squares on the right, which was undercooling on the ground using electromagnetic levitation, and the red diamonds triangles on the left, which was the electrostatic levitator with Marangoni convection, I would not have had any clue at all what was going on in this system. But because we went to space, we were able to fill in different shear levels and therefore come up with a theory. And the theory that we decided to use was well-known Reed-Shockley dislocation energy theory, which states that the energy that's contained within a system is equal to a complex function of theta and some constants, where theta is low-angle grain boundaries, and those low-angle grain boundaries are proportional to the number of defects in the sample. So we have a relationship that relates energy to the concentration of defects. And so when I do that complex function, if you do some mathematical manipulations, you realize that the free energy over the shear rate plotted as a function of the logarithm of the shear rate should yield you a straight line, and it does. Therefore, we feel very confident that this is a good way of representing what's going on in this system. So now we know mathematically how to predict the effect of convection, that free energy is conserved, and mathematically we know how to predict how much convection influences that free energy. So I'd like to do this in dimensionless form so that I can compare different alloy systems. And so what I do is I use as the reference state that baseline condition, if there had been just barely enough undercooling so that we saw the double recoalescence. What is the longest possible delay time, i.e. the smallest possible energy, the free energy involved in the system. So that's the reference state. So I take my experimental value and divide it by the reference values for both the delay time and the driving force. And again, what I'll expect, the thing I'd like you to take from this slide is that thing with the black square around it. I would expect that the dimensionless incubation time number should be inversely proportional to the driving force to the -4th. When I do that, the data, as I showed you originally on the left, which was delay time as a function of undercooling, collapses down to a single line with a slope of -4.06 with a little bit of error in there. So that is really good verification of our theory. We were very happy with the space results. You can see that they really filled in the necessary information for us to understand what was going on in this one system. Now, this was done again for one alloy, and we're hoping to do it for additional alloys. This particular alloy is a 60% iron, 20% chromium, 20% nickel alloy. So why don't we also try to plot the ground-based data that we have for the other members of this alloy system? That plot on the left, you can see, that's what the data looks like. And when we use our new theory, again, this is with absolutely no adjustable parameters. That once you've set things for the 60-20-20 alloy, you use those same parameters for the evaluation of the others. And you see that the entire family collapses down to a single line. So we think we really understand how to control transformations in this alloy system, and based on the undercooling and the convection. So the science conclusions are: incubation delay in this entire austenitic steel family is modeled successfully using this damage-modified classical nucleation theory. We show that this retained damage has two sources, undercooling and convection. And the ESL data gives us a clue on how to look at what goes on for undercooling. The EML on ground is augmented by the EML in space, because we can now set a wide range of convection conditions and understand how damage is modeled due to convection in the sample. Therefore, the Reed-Shockley approach was worked out as a very good way to estimate how much energy is being put in as a function of how much droplet shear occurs. And lastly, space data has been just really important in order for us to fill in the gaps between laminar and turbulent conditions for this alloy system. So I'd like to acknowledge my many collaborators on this system. On this particular alloy, the sample was run by the University of Ulm. They did a lot of thermophysical property work. Jaing Li and Bob Hires helped us out with the modeling activities, and of course NASA Marshall has been instrumental in running a lot of this ground-based research. I'd like to acknowledge NASA under the grants that I have on this project. I'd like to close with just an observation. So why should NASA care, right? I mean, what's the big deal about convection in steels? Well, when you're in space and you go to Mars and you break a tool, what do? That's a picture there of my toolbox. It's got a little red circle there. Somehow, I don't know how. I don't know how it's physically possible, but I lost one of the. You all gone through this. I swear it was my children, but it probably wasn't. My children all deny it, and I believe them, so I must have lost it. And what I can do? I can go to the hardware store and I can buy another one. It's about 10 years since I lost that, so I haven't. But what do you do when you're out in space? You can't bring 5 or 6 of these kits just to make sure that the one that you lose you can replace. You really can't. So what you do is you make your own. And this is something known as additive manufacturing, also known as 3D printing. For some reason, there it goes. So this is I'm sure many of you already know about additive manufacturing, but I thought I'd just put this up here. This happens to be a plastic part that we made in our lab. And my grad student thought this was the best thing I ever asked him to do. He made about 5 of these because he wanted to give them as Christmas presents. So what you do is you go ahead and you print up a part, and this prints up a crescent wrench that's working. I mean, you take it out of the machine and you can turn the knob and the crescent wrench works perfectly. The problem is I don't want plastic parts in space necessarily. I'd like to be able to do this in metal. And Marshall's doing a lot of work with additive manufacturing in order to produce not only parts that can be used on orbit and during colonization operations, but also for this novel hardware like new rocket systems and being able to make very complicated parts with internal structures that are astonishing. And we don't know what the effect of low gravity is going to be on the production of these parts. Have parts been made in space? Yes, they have successfully made parts like this. But the question is, how do you optimize? How do you know where the strengths are? Where are the defects in these parts? We need to be able to do mathematical modeling that is accurate and predictive so that we can control the properties of these materials and understand how to make quality parts. And that's the goal of this research that we're doing today. Finding out, using using these techniques to measure thermophysical properties and to evaluate, therefore, to be able to put those good properties into mathematical models so that we really understand how something's going to react. I'm working also in collaboration with the Japanese Space Agency on a project using their electrostatic levitation furnace that they have on orbit. You'll be hearing a talk later today. One of their representatives is here to give us some insight into this exciting new facility. And I just want to say that's one thing, is the thermophysical properties are important, but also how's consolidation of these metals? The way you do this with metal is you spray powder down and then you hit it with a laser. Therefore, surface tension is going to be huge. And the influence of oxygen on the surface of the powders used is going to be huge. How do you keep powder down so that it stays in place for the laser to hit it? These are all questions that are going to have to be answered by new research activities. So although I'm a little bit short on time here, I thank you for your attention, and I welcome any questions.
We have time for a couple questions. That was a great talk. But I'm curious about when you talk about damaging a undercooled liquid, you're implying that there's some structure in the liquid. And is there any direct evidence for structure in undercooled metals?
I anticipated your question. It's not damage in the liquid. Remember, what we've done is we've formed a metastable phase. That metastable phase has damage that's induced into it. And so you have, for instance, here shows on, this is a cartoon of a perfect structure. And in order to fit in this metastable phase, you have to have significant diffusion. And in fact, you have to kick some of the molecules out. But if you have a bunch of holes, it's a lot easier to fit in the growing nuclei. In fact, the growing nuclei helps to relieve some of the strain, and that adds energy to the system. So it's the metastable phase where the damage has been retained, and it's not in the liquid itself. Yes, the liquid shear. The liquid shear causes damage within the growing metastable phase, and that damage is retained. And then you asked, the second part of your question was, is there any physical evidence of that? We have, when we first did these experiments, we were very surprised that it worked so well. That's the first comment. I'm sure all of you who do space experiments know that feeling. It's when it works, you're really, really happy. And then we had that exact same thought was, oh God, now we've got to prove it. And so we've initiated some different experiments. The first was just serendipitous, that we had run a second sample that's titanium aluminum, and it also goes through transformations, but we were noticing that the transformation, you could actually see healing and grain refinement of the primary phase. So we were seeing this same effect, but it wasn't a phase transformation from one phase to another. It was actually recovery. It was growth of new grains, a recrystallization of new grains of the same phase. And so that was exciting. But that's indirect proof. The direct proof is we've now taken samples down to Argonne, and we've run diffraction experiments where we put the X-rays through the sample real time. The technology has just recently advanced where we can actually get data. In the past, we could only get data at 5 hertz, and that wasn't good enough to see these transformations. And the new technology is getting us to 250 hertz. And so now we can actually see these transformations. And so you can actually see those of you who've done diffractometry know that an individual crystal plane will diffract X-rays along with the preferential Bragg angle, and the spreading of the X-rays around the peak. So if it's a pure material that's absolutely pure, that has no defects in it, you'll get a beautiful straight line, one angle. for the Bragg angle. But if you've got a bunch of bent planes due to dislocations and due to defects, that spike will broaden out. And the amount that the peak broadens out is proportional to the damage, and we are seeing that effect. So we actually can see this real time during solidification, showing that the damage really is there.
What thermal properties you have used to calculate the excess thermal energy or any other parameter that you have presented in the metastable condition?
So again, I anticipated that one. we have a bunch of properties that we need to do the modeling activity. These are listed over there on the left. We need to know the concentrations, the melting points, the packing, the heat of fusion, melting temperatures, and what we do is it's very difficult to do the calculations, as you note, on a metastable phase. And so what we utilize is some canned programs called ThermoCalc. And ThermoCalc gives us predictions of what the thermal properties are. And these are listed in this table, and anything that's green means there really wasn't any difference between the alloys, and anything that's orange means that was a big number compared to everybody else, whereas the yellow is a little number. And you can see for all these alloys, it bounces around. It's not like there was any clear trend here. And yet, we still ended up with that great agreement. So we utilize thermophysical property models in order to come up with the properties that you asked about.
Okay.
Well, thank you, Doug, for that wonderful talk, and we'll go ahead and move on next. Thanks a lot, Doug. Thank you. Our next speaker is Dr. Chris Wolverton, who is a professor in the Department of Botany and Microbiology at Ohio Wesleyan University, and he'll be giving his talk on plant responses across a continuum of gravity treatment. Dr. Wolverton, welcome, Dr. Wolverton.
Thanks, Kevin. I need to step out for a second. I'll be right back.
Sure. Excellent. Well, thank you. Thanks so much for the opportunity to share some of the results of our spaceflight experiment working with plant roots. I'm thrilled to be here. And I want to start off by just acknowledging the incredible team effort that goes into a project like this. I know many of you are familiar with that, having done your own flight projects, but just to note, this is my team at Ohio Wesleyan, which is an undergraduate-only institution. I've had some 18 or 20 students participate in ground preparations for the project, and two of them are here today, Lexi and Claudia, and they'll be presenting a poster on some follow-up work in collaboration with Sarah Wyatt and Al Myers at OU. Of course, the entire NASA team, we used an ESA facility, and so the ESA team, and in particular the Norwegian User Support and Operations Center, the ISS crew, the ISS team, the office, it's just amazing to see a project like this, as Doug said, actually work. And so that's incredibly exciting. I hope you'll find our results interesting as well. So there's just a couple, just to put some faces with the names. My team at the lab at the university, Nathan Modonich there with Guoxing Sun at Ames Research Center, and the Ames Center, the hardware was developed there. And so I just want to start with acknowledging the incredible support to do this project. So the first thing to note is that the shape of plants is determined by gravity. There's never been a plant that's grown, other than those that we've sent to low Earth orbit, that hasn't developed in 1G. And as we take a walk through the woods this fall, we can see the influence of gravity in the very vertical growth of shoots, of stems. We can see, in fact, gravity's influence even on branches. Those organs that are non-vertical are still being impacted by the constant 1G environment, sensing that, producing reaction wood to reinforce their growth so that the branch doesn't snap off. So you get a sense when you look out at the woods, not only of how lovely it is in the fall in the Midwest, but also I see angles, right? And these angles are in large part informed by the plant's response to gravity. Of course, the light environment and neighbors and competition and the quality and fluence rate of light is also determining shape, but gravity is this constant, it's persistent and influencing the shape of roots, not only above ground, but also below ground. And what my lab primarily focuses on is roots, and here you're looking at just a cross-section of root shapes, root systems, and their architecture that's also influenced by gravity. And so the branch angles of those roots have tremendous implications for a root system's ability to mine the soil for nutrients, to explore for water. And those angles that roots attain are in large part a combination of both genetic and environmental, primarily gravity, because here under the ground, we're not particularly influenced by other cues like light. Cues like touch and obstacle avoidance and moisture gradients take the place of what light does above ground, we think. And so just keep in mind, as you sit in your chair, you know, hop up just a little bit and you can feel gravity. It's easy to forget that it's constantly there. But as we think about gravity and the gravity responses in plants, it's easy to miss that plants are so sensitive and responsive to gravity because it's happening slowly. And here in this time lapse, this kind of helps to bring it to the forefront. Let me play that again. Oops, not that way, but that way. We get a sense in the time-lapse video of over the course of 4 hours, these roots can reorient almost perfectly vertical again, and the shoots, the hypocaudal parts, are also reorienting. I also like to start with this slide near the beginning because it points out how we're growing plants in our spaceflight experiment. These are Arabidopsis seedlings. Each one of those little boxes you see on the membrane is a 3 millimeter square, and so it gives you a sense of scale. But our roots are elongating down the surface of these membranes, and we're primarily going to be interested in capturing images and measuring angles of those organs as they're responding to gravity. So you can see it's not a nervous system muscle type rapid response, but it's a response nonetheless that's evolved over hundreds of millions of years to orient plants in the gravitational field. So, of course, plant shape on the Earth is determined by gravity, and as we leave the Earth, as we do exploration, the shape of plants that we grow to sustain the crew for vitamin and micronutrient needs, for potentially larger calorie food crops in longer duration missions, the shapes of those plants on the Moon and Mars is going to be determined by the gravity environment on those bodies. Gravity is not something you can particularly pack up in a suitcase and take with you. And so presumably we will be growing plants on these bodies with no augmented gravity to speak of. So the first question, just by way of background, is how do plants sense gravity? And we know quite a bit, going back over 100 years to this question. We know that the tip of the root, the apex of the root there, that you can see, there's sort of some shadowy figures that are highlighted there in this differential interference contrast image of an Arabidopsis root tip. And those play a critical role. We know from fellow ASGSR investigators who have worked out which of those cells, those cell tiers, are actually doing or are most sensitive to gravity. But the general theory is that in those cells, in the tip of the root, exist starch-filled plastids. And these are sister organelles of the chloroplast that are photosynthetic and green. They're filled with starch in these specialized gravity sensing cells at the tip of the root. And when the root either encounters an obstacle and is displaced from a vertical growth trajectory, or we sneak up behind it in the lab and tip it on its side, what happens is that those starch-filled plastids sediment. And this sedimentation as they drop to the lowest point in each of those successive cells, is indicating position. It's indicating orientation in some way to the organism, which results in a differential growth process. And I'd like to also point out that these kinds of responses, the growth of plants in response or in response to a stimulation like this, is coordinated across thousands and thousands of cells that span the length, in the case of Arabidopsis roots, of almost a millimeter. And so this is not a bending phenomenon. It's not purely a physical phenomenon. This is a controlled differential cell elongation process. And so the positional information that's communicated or transduced to the cell is a direct result of the sedimentation process that we're watching here, and then goes on to coordinate a signal transduction pathway and the transport of important hormones that regulate cell expansion in the growing zone. So we've got a kind of a classic example here of a process that's happening in one location and is having an influence on another location, a kind of classical hormone response system. We know that these stabilists that are filled with starch are extremely critical for gravity sensing, because mutants that lack starch biosynthesis, and this one in particular has been studied by John Kiss and collaborators for many years, the absence of starch leads to a reduced ability of the plant to respond to gravity. And what that looks like, here we can see two root caps, and the wild type root on the left is full of starch. When we stain it with iodine, potassium iodide, we see these dark figures. This is just kind of a quick and dirty benchtop microscope image. And the root on the right is deficient in an enzyme called phosphoglucomutase, the first step in starch biosynthesis. And so it lacks starch-filled the plastids are still there, but they're empty sacs. They're not filled with starch, and therefore they don't sediment. One of the interesting things about these mutants is that when we take very high resolution time-lapse images, which is what my lab has focused on for 20 some years, here what you're looking at is a vertical rotating stage that we can place growing roots on and take images with the camera that's at the top of the image here, and in real time trace the shape of those roots, determine the angles of various regions of the root, and get a good sense of how quickly those roots respond to being reoriented in the gravitational field. And when we tip a root on its side and just allow it to respond, that response takes place in wild type roots, as you can see. By about 4 hours, that wild type root has gone from approximately horizontal to approximately vertical. So again, it's a slow response, but it's responding nonetheless. The starchless roots take quite a bit longer. They get back to vertical, but they do so much more slowly than wild type roots. So what this is telling us is that yes, those plastids are critical for full gravity sensing, but this also suggests that they're not the only story, that there could be some other story. And so what we've done in my lab is we've taken advantage of that vertical rotating stage that I showed you. And as we take images of the root every 30 seconds or so, determine the angle, we can tell that vertical rotating stage to hold the root tip constant for hours and hours and hours at some prescribed angle. And what that gives us is a root that looks like what you see there. This is a time-lapse sequence of images that were collected during one of these responses of a wild type root. And so we think of this as kind of a gravity clamp system where we're holding the root's orientation in the gravity field constant, and we're allowing it to express its as much differential growth as it can. And what we find in wild type roots is that rate of response, as we hold the root at fixed angles from 30 degrees to 60 degrees to 90 degrees, as we step that up, what we find in wild type roots is that the rate of response goes up and up and up to a maximum at 90 degrees. And this has been observed not using the fancy computer-based feedback system, but for many years, and termed the sine law in gravitational research in plants. So you can see that upper trace on the figure on the right is representing the wild type response. An increase in stimulation angle leads to an increase in the rate of response. And here, because we're able to hold the root at a constant stimulus and observe its output, that output is extremely constant because the tip angle is not changing where all the sensing seems to be happening. The interesting thing is when we look at starchless roots, those roots, they show no dependence of their output response on the stimulation angle. And this is really cool. This is really interesting, something so different than the wild type conditions. You remove these sedimenting statoliths from the system and you get gravitational responses, but there's no dependence on angle. And so this is suggesting that those statoliths are contributing the position sensing, but yet there's still some way for these roots to detect that they're oriented away from vertical even without that position sensor. Some potentially second system, which is, we need to keep in mind in wild type roots, active in not only that second system, but also the statolith-based system. In starchless roots, they're lacking the statolith sedimentation and they only have that other system. And so we're trying to determine what those systems, if there are, in fact, two separable systems. So we can conceive of one kind of output system that controls, it sort of generates one signal for differential growth. And whether you're in wild type roots or starchless roots, what the plastids are doing is just potentiating the output from that single system. And so on the left, we've got kind of the example of some statoliths that are sedimenting on some as yet unknown receptor that's detecting their position and producing some singular output that defines the rate of response. In starchless roots, you don't get those empty plastids sedimenting, and so they're producing a response output, but that's not potentiated relative to the stimulus angle. This is one hypothesis. A second hypothesis for what's going on is what if those statoliths represent one type of output? And so that's shown there with the red arrow on the bottom left panel. In wild type roots where you have functional statoliths, you're getting output from that statolith system to control that differential growth response, and also whatever secondary sensory system, some background sensory system, which is less than the statolith system. In starchless roots, all you're getting is that secondary output, that sort of background output that's not potentiated by statoliths. So we'd like to be able to distinguish between these two systems, whether there are in fact two systems, and what kinds of differences in control they might represent. And so our flight experiment was focused on these key questions. First of all, can we distinguish starchless and wild type roots, statolith, intact, and no statolith root systems based on supplying different amounts of g-force or acceleration? And to do that experiment, the best place to do it is on the station. And so getting out of Earth's constant 1G background, sending them to the ISS, and adding some gravity back and asking, can these two different systems, are they activated at the same acceleration force, or are there differences in their activation? So that's the first question. The second question is, does the rate of response vary depending on G, right? So instead of depending on the angle of the root tip, which is what I've just talked about, the experiments we focused on the ground, if we can vary gravity instead of the angle, and ask, do we get a similar difference in output as what we see when we vary angle? And so that's the second question there. And then the third is really to go back to this bigger question. Do these two potentially different perception systems control one common growth regulatory output, or potentially two or more growth regulatory stages? And so to do this experiment, as I mentioned, we've used the European modular cultivation system, which has now been retired from station. We used it with NASA Ames specific hardware that they developed with Dr. Kiss to do gravity experiments by adding, the EMCS contains a centrifuge, in fact, 2 centrifuge rotors. And these specialized hardware that you see shown on the left, that's about the size of a tissue box. And that hardware contains within it individual seed cassettes. And those cassettes, one of the neat things about this hardware is those cassettes, they're arrayed along the centrifuge arm. So if you look at the diagram on the right of the kind of centrifuge rotor mock-up, each one of those cassettes holds about 14 seedlings for us. And because they're a different radial distance along that centrifuge arm, the seedlings in those cassettes experience slightly different g-force, roughly twice as much on the outside as on the inside as it's spinning. And so what you can see is we've applied in this experiment a number of fractional gravity all the way up from 3/1000ths of a g, as slow as this rotor will turn, all the way up to Earth 1G control on orbit, and ask this question: what's the threshold like for these two different types of roots, and do they show differences in their output responses? So here is our hardware floating in low Earth orbit with astronaut Akaba here. You can see the bag full of these experiment containers on station, and the EMCS, one of the doors is open there on the right side, where the crew can simply install these, or could install these into the EMCS, and commands from the ground would activate and hydrate the seedlings to initiate the experiment. So very flexible design to do these kinds of projects. We could launch dormant dry seeds and conduct the experiment as crew time permitted. I should take a pause here and indicate, even though the hardware's been retired, we experienced some technical problems with the hardware during the experiment, and just once again to point out how grateful I am to NASA and to ESA for working through those and getting us almost all of our science back. So I'm really grateful for that. It would have been probably easy to sort of cut their losses and not conduct the experiment, but we've got some great data to share because of their great effort. So the timeline, the kind of experiment, NASA really loves these kind of timeline charts, Gantt charts, or what have you, to indicate the kinds of treatments that are going to be supplied during orbit. I think it's a little bit easier in this case, just to point out that what we're doing is we're going to grow our seedlings after we hydrate. We're going to grow them up for a while. So they're going to be 3 days old. And those seedlings, while they're growing, they're going to experience the equivalent of on average 1G. So we're going to grow them so they grow nice and straight. They're growing under white light from above. So we get nice growth to start with. And then we turn off gravity. And I love to be able to say that. We're in the ISS, and so we can turn the rotor off. Now the roots are going to experience microgravity, and we're going to turn a light on from the side. And this blue light, we hoped, would induce curvature of the roots away from the former gravity vector. And normally when we do gravity experiments in the lab, as I indicated before, we can sneak up behind the Petri dish and tip it on its side. We can't do that on the rotor of this centrifuge. And so to get tissue in a stimulated orientation, we're really doing this blue light treatment as a preparative step to get those root tips in the angle that we want them, so that then we can turn a fractional amount of gravity back on and ask whether the plant can feel that gravitational signal. And that period lasted where we allowed the roots to experience whatever prescribed gravity rotation treatment. That was a 36-hour treatment. And throughout this period, the design of the system allows us to capture time-lapse images of the seedlings as they're growing on seed cassettes and responding. And so that was our intended effect of the treatment. The actual treatment, so these are some spaceflight samples shown here. Just for example, you can see it worked. So this is, as Doug shared, it's always amazing and exciting. The treatments work. So you can see on the left some wild type plants that had experienced 12 hours of that unilateral blue light. The blue light was coming from the left of that photo, and so the roots are growing toward me, and they're highly, in this case, they're primed to be stimulated with that different gravity treatment. And that's what you see for that same membrane 36 hours later, under 1/10 of 1G. That's what the roots look like. And so you can see here a very clear reorientation toward the bottom, downward in this case, is along the gravity vector that's supplied by the centrifuge. So that brings us to our data, our questions. What is the threshold for response in roots in Arabidopsis? And to kind of start at the end, I'll say that Arabidopsis roots appear to respond to as little of a gravity force as we could supply in this experiment. So the EMCS would allow us to stimulate at as little as 3/1000ths of a G. And as you can see here, the lower left shows the growth of those roots after that 3/1000ths of a G treatment 36 hours later, aligning along that gravitational vector. And so extremely sensitive to that supplied gravity force. And I'm going to walk through our results to show sort of, instead of just cherry picking the best image, I'll walk through and show you all our data here in a second. On the right is our starchless mutants, and those seem to require about 100 times more acceleration to activate their response. And so a dramatic difference when those roots are lacking those statilysts to begin responding to gravity. So let's walk through. This is sort of, as I said, starting with the conclusion. So let me walk through for this part how we get there. The first way we test this is we just, an army of us looked at each root that we could score, and we said, is this responding or not? It's a 1 or 0 kind of a binary attribution that we apply to all of our almost 1,100 roots that grew. And as you can see here, in no treatment were the wild type roots responding at lower than 50% response rate. There is a bit of a trailing off as you approach that lowest treatment, but at least 50% or more of the wild type roots were responding at as low as we could get. I'm not sure how good your eyes are, but I've got numbers on top of those just to give you a kind of sample size idea as well. So obviously, we can't make strong conclusions when we have very few roots at any particular fractional G treatment. But compared to the starchless roots, which really don't start to get up to that 50% line or above until at least a tenth, more like 2/10 of a G, or even slightly above. We're not up there consistently until up around 0.3 G treatment. So that's one way to look. We've got 4 other ways that we've analyzed response or no response to get at this question of threshold. The second approach that we've used is let's just look at the tip angles of all of our roots. So here what you're looking at is from the left, the lowest G treatment, over to the right, the highest G treatment. What's happening to the change in tip angle from its starting point? This is kind of per individual root and then aggregated. How much tip change is produced at that G level? And as you can see here, the orange bars indicate wild type and the green bars indicate starchless. And we don't start to see consistent average increases or changes in tip angle in the green bars until we're, again, up in that same region, somewhere up around 0.1, 0.2 G. Whereas the tip angle change in the wild type roots starts as low as we go. It's less than what we get up around that 1/10 of a G, but the tip angles on average are still showing some response to that G treatment. So that's a second way. A third way that we can analyze this is let's apply some filtering to the data set, and let's define what we mean by responder to gravity as those roots that have a starting angle above 20 degrees. So that's the minimum on the ground at 1 G that a root will initiate a gravity response. It has to be gravity stimulated, and that angle is somewhere around 20 degrees. So roots that are above 20 degrees, they achieved roughly what we'll call vertical, which is somewhere in that plus or minus 30 degree range. So they got back to vertical. We'll call that a responder trait. And we'll say that they generated at least 20 degrees of total tip change. So they were going in the right direction, and that it was at least 20 degrees worth of change. And of course, the other parameter we have to include is that some of these, as we score them, they just stop growing. And so we also have a parameter in there that captures was it growing or not. And so we're going to filter this data set. What you can see is almost all of our PGM examples, they drop out. You don't see any green on the left side of this plot. And that means that those, all of those seedlings, some 500 or so of them, they're not meeting those criteria that we've set. And so they weren't gravity responders by this definition. On the contrary, for wild type roots, for the most part, we get that continuous example of response all the way up, all the way across our G treatments. So that's another way to determine response. A final way, well, third, fourth, what are we up to now? Fourth way to determine response is let's just throw all of our final tip angles into circular histograms. So these are histograms, they're binned by number of observation, So each of those little black bars is representing some category, plus or minus 15, 15 to 30, 30 to 45 in either direction. What we would expect at 1G, which is shown on the top right for wild type, is that we should be seeing roots that are roughly vertical, right? So that's what vertical looks like, that example on the top right. Over on the far left for wild type, not everything is vertical, but we've got this tendency toward vertical, even at our lowest treatment levels. And here what I've done is, rather than making 25 or 24, I guess 25 of these individual histograms, I've lumped into just 5 treatment groups, okay? So these are ranges that I'm gonna work with for the rest of the talk. So we've got the histogram that also shows the same thing. We don't start to see that orientation around vertical in starchless roots. We've got a little hint of it there in that middle group on the lower row. You can see we get a bin of starchless roots that are starting toward vertical, probably right around that 0.1. That, by the way, the middle grouping is roughly moon gravity. The next one to the right goes up to Mars gravity, and then our 1G is on the far right here, okay? A final way we can ask this question is, what's the comparison of the total tip angle change? And again, we're gonna stick with the groupings, but in this kind of plot, our 1G is on the far left. And that's because I wanna compare 1G as our control, and this estimation statistics package, the way it's designed to work is to put your control on the left, basically. And then what we're looking at is, along the top is all of our data. So each point represents an individual root. And you can see the black bars next to that red grouping. Those black bars represent the mean, where the notch is, and the standard deviation for that population. All of our roots, in other words, showed a total tip angle change at 0.5 to 1G of an average of 82 degrees. That's responding, right? And as we move across this from left to right, we're getting less and less G, all the way over to the far right, where we see in wild type roots, they're generating less total tip angle change. And that difference is reflected in the bottom part. So that what you're looking at on the bottom of these graphs is a 95% confidence interval histogram that's representing the effect size, right? So that black bar on the bottom chart is representing the mean change from the top, and that's fixed at your control group. So the 0 difference represents the control. For the second from the left grouping, the blue grouping, there's no difference at that level between the 1G and that treatment, okay? So we're gonna be using these plots. I thought I'd take a minute to explain what you're looking at here. This, I think, is a little bit fairer and more honest way to look at the data, rather than just doing t-tests and saying difference or no difference. This gives us a sense of the size of the effect that we're talking about. So this is the wild type tip angle change, all of our data, right? So the filter is all growing roots. You can see that on the bottom left. When we look at the same thing for PGM, what we find in looking at all of the growing roots is that basically those first 3 categories from the right, there's no tip angle change. Again, consistent with this sort of the other 4 methods that we've used to identify threshold in PGM. There's no significant tip angle change. We start to see some tip angle change in that second grouping from the left. And then when we're back up to 1G treatment, we've got significant tip angle change. Again, this is all of our roots. And so what we can say is, for the perception threshold, it's not the same in wild type and starchless roots. We see a difference. It requires much more force to activate the response in starchless roots. Okay, second question is, does the rate vary, or the total response vary as a function of G? And so what we're looking at here is, you can see these differences when we look at wild type roots that are stimulated at the very low 3/1000s compared to about 0.5 G. What we find is this interesting difference in shape. And you can see this, the curvature, the geometric curvature in this case differs. The radius of curvature is much sharper or smaller in the roots on the right than it is on the left. And what we'd like to be able to do is to get at response, total response or response rate as a function of either G force or dose. And so I just showed you this plot, and this is throwing all of our roots in and asking how much tip angle change, and we use this to get at the question of threshold. If we filter this data set, into only those roots that are responding. In other words, if we take all of our data and we filter by they're growing, they're stimulated at least 75 degrees. So they're starting at a highly stimulated angle, and they're showing at least some tip angle change that's positive. What we find with wild type roots is we find some kind of dose or force effect on the total response that's generated over that 36 hours as we increase up from the right to left in this plot, as we increase the amount of G that's supplied. So we've got a significant difference over there on the right. You can see our confidence intervals don't overlap with the control line on the bottom at all. For those first two categories, they just start to at the moon level. They're almost completely overlapped. In other words, there's no difference in wild type in the total response when we supply either the moon G or 1G. When we do the same treatment in starchless roots, let me go back. So this is what we had before. This is all of our roots. When we filter this using the same parameter, what we find is, interestingly, at the point where they're consistently above threshold in their response, so only in those two categories, starting at 0.2 up to 1.0 G, what we find is that there's actually doesn't appear to be any difference in total tip angle change for these roots. So this suggests actually that statoliths are potentiating the influence of variable gravity on the response. In other words, this difference that we see as our wild type roots climb from the lower treatment up to the greater G treatment, that difference must be mediated by the effect of statoliths. And when we lack those statoliths, what we find is that the response appears to be either on or not, that it's not responsive, it's not potentiated based on the dose of gravity. So coming back to that question, does the response rate vary depending on G-force? We don't have the best data, which would be kind of minute-to-minute data on the rate of response as a function of G, but we have this total tip angle change that we can use to get at least partially this question. So returning to our model, I think what we've seen here is that these statoliths potentiating, they're sensitizing the root's response to gravity, because without them, those starchless roots are far less sensitive to gravity. And they also seem to be potentiating that difference in total tip angle change, such that starchless roots appear to be either on or off, which is remarkably consistent with their responses on the ground to variable tip angle. Remember, those responses were either on or off. They don't show any change. And we seem to be seeing a similar thing here with the spaceflight variable G application. The response of starchless roots seems to be either on or off, which I think probably argues more for the latter model, the lower model that's reflected here, rather than the upper. So let's move on and talk just briefly with the time I have left about phototropism as another means to dissect these gravity responses. So the phototropic response looks like this. Whoops, let's go back. Does that movie play? No? Can you start that movie from back there? Nope, maybe not. Okay. So my interpretive dance version of the movie is that these roots respond consistently away from the light, so they're going to be curving toward me minute by minute, slowly, until they're perfectly vertical. We sort of saw the results of that, and there it is. It's just like I said it'd be. Okay, so that response is happening in 0G. That's in microgravity, and so we're getting that really robust root curvature away from the light, shoot curvature toward the light. This is 1 image per hour played back 2 per second. So what we find after that 12-hour treatment is, again, with our circular histogram comparison, our roots are more or less horizontal, right, in both genotypes. They're more or less horizontal. This is in microgravity. When we're on the Earth, what we find is that they're competing, right? There's competition between gravity and light, and so gravity's pulling the roots down, light is pushing them to the side, and so in wild-type roots, what we find is that they land somewhere around 40 or 42 or 45 degrees, the starchless roots at a higher angle. And this makes sense. The way we can think about this, though, in terms of gravity sensing, right? We're using light as a challenge against gravity in this case. And what we find is, after 12 hours in microgravity, there's no difference between starchless and wild-type root orientations. That's what the analysis on the left shows. On the right, on the ground, what we find is that there is a significant difference, and that effect size is about 15 degrees. In other words, starchless roots are about 15 degrees farther away from gravity than wild-type roots. And that seems like easy to miss in passing, but what we're talking about there is that 15 degrees is imparted. That is the influence of statoliths on this gravity sensing system. That's where the difference comes from. The fact that there's no difference on the left is our control. That says that these roots respond they respond exactly the same when you take gravity out of the equation. When gravity is there and you have statoliths, you get 15 degrees more response to gravity. When you have no statoliths, that is missing. All that's there is the background, the alternative system. As another control, we did this same experiment on flight where we applied gravity and light for 12 hours. And what did we find? We found exactly the same difference in the EMCS centrifuge at 1G as we find on the Earth at 1G, right? The difference between starchless and wild-type roots. This is a great control for the use of the EMCS centrifuge as an artificial gravity environment, in other words. Okay, the final bit of our work is not completed. Well, it's getting there, but each of these samples is going to be, has already been isolated. Its RNA has been isolated, and we'll be doing RNA-seq at 12 different G treatment levels. So across this entire continuum of gravity treatments, we'll have a transcriptome to compare the influence of gravity at that G force all the way up to 1G. We're looking by organ type, by genotype, our 2 genotypes, and then plenty of replication. So this is an augmentation from GeneLab with Sarah Wyatt and Al Myers. We're eager to get those data back and get at some of these more mechanistic questions that I think we'll be able to bring to bear and overlay on top of our physiological response data. So this is basically what I've spent the last 35 minutes saying. You can look that over. What we're doing right now is Al has just completed the RNA extractions, and those are now off for library prep and sequencing. And next year, we hope to be able to overlay, as I said, that story on what we've been talking about right now. So to conclude, our threshold for gravity response in the statolith pathway is at the minimum we could apply in this instrument. The non-statolith sensing pathway has a much lower sensitivity and doesn't appear to be potentiated by the change in gravity. The statoliths sensitize the root to gravity, and they may also be potentiating that gravity force or at least gravity dose. So with that, I'm out of time, and I'll take any questions. Thank you.
We have time for a couple questions.
David.
Thank you, Chris. That's a remarkable story.
Thanks.
I was shocked when I saw the low level of the thresholds 35 years ago. God, can it be that long? Alan Brown and I had knockdown, drag out fights about the threshold. And he must be smiling some.
Well, what's remarkable to add to that is that what we're seeing is in fact consistent with some early work in the 60s, Jane Shen Miller and others kind of extrapolated down to this low of a G force, or at least a G dose that should activate gravity sensing. In fact, I think their number was like somewhere around 2 to 4 1,000. So we are not seeing anything that differs from that.
Well, you know, I don't know. I wasn't convinced of the data at the time. We were on the Gravitational Research Facility Science Working Group, and we were arguing with the engineers about what level of vibration they needed to meet to tease out the difference between the centrifugal and the vibration. So I'll ask you the same question. Have you actually looked at the vibration? And you know, it's linear acceleration, just like gravity. It's just a different frequency.
Right.
Have you looked at that as a potential contaminator in the low levels? And
yeah, it's a really good question. I think it would be. I think it would rise to that level. Obviously, depending on all kinds of factors, like what, you know, the vector of that, you know, frequency or vibration or disturbance or whatever. We've not, and I don't have a good system or a good, you know, a good model to test that in. We have done, you know, we've controlled for that as best we can in some of our ground pilot work, where, you know, when we wanted to collect RNA from gravity-stimulated roots, rather than just leaving the batch alone, we treated it the same way. You know, we pick it up and set it back down, and instead of picking it up, rotating it and set it back. So we're trying to control a little bit, but measuring the effect of that, no.
Well, and you know, I'll just another comment. I mean, it's remarkable what we can learn about these things by exploiting the capabilities that we have in space flight.
Absolutely.
I'm sure Craig is sitting there wondering, does this have to do with growing plants on the moon? Do you have a practical side to your conclusions?
Absolutely. I mean, the practical that I often give, I'm in the Midwest, and so I've given this talk at several public libraries and you know, talked to farmers about practical applications on Earth. But the entire root system is gravity driven. Obviously, the flow of water and minerals in the soil is driven by you know, capillary flow, but predominantly by gravity and by convection. So the practical in terms of the root is, once we understand what some of the molecular components that are controlling either or both of these systems is a little bit better, then we can, for example, I could imagine using plants that have either knocked in or knocked out components that sensitize or desensitize roots. You know, one hypothesis that we're starting to see repeated over and over is that spaceflight and reduced gravity environments are perceived as a stress in plants. And so maybe it makes sense to actually make them less sensitive to gravity and allow them to explore a growth substrate for water and minerals, and just ignore the fact that they can't detect gravity. So that would be the one big practical thing. Great, thanks. Ray.
Yeah, Chris, great talk, thanks. In the centrifugation, did you run that for 36 hours? Is that correct?
That's right.
And so were you able to see when the bending responses first began with time-lapse photography to kind of get a minimum presentation?
Yeah, we have some data, but it's limited by the fact that the camera, we wanted to do it completely in the dark during that 36 hours. And so the camera system has an infrared light built into it, but it shines from the front. And so most of our sample has significant glare on the surface of the seed cassette. So we can't see what's happening on all of them. We do have some where the angle is just right for that lighting. And so I don't have quantitative data yet. We're trying to kind of clean up some of them and get a data set. We'll only have it at certain G levels. It won't be a kind of population level like we've got here. But that's a great question, and would go some way toward teasing apart. I've used this phrase, gravity force and gravity dose. And that kind of behind the scenes, what I mean by that is, if you imagine our lowest treatment, those are actually giving a gravity dose of about 30 minutes equivalent of 1 G on the Earth. And so that 3/1000ths, if you kind of do the multiplying through, the dose of gravity is roughly equivalent to about that. Actually, it's even, maybe at that treatment, it may even be as little as 15 minutes. So looking across, using the time of response, if we could see it, to answer the question of how much gravity as a dose is required, would be a great way to use the data here. We're trying to pull it out, but it's not there yet. Thanks, Ray.
Here, please. Well, excellent talk, Chris.
Thanks.
Thank you very much. Well, I have, the discussion could be highly interesting. A couple of small things. On the one hand, I would like to maybe to differentiate between the threshold for sensing gravity by the satellite system from the effects of the gravitational stress. Because, for example, at the moon gravity, we have performed experiments in simulating the moon gravity, and we have strong effects at the level of the cellular and molecular physiology of the, for example, of the meristematic cells.
Right, of division,
so,
right.
You can show that really there is a perception of these very low levels of gravity at the roots. In fact,
and you found that at about 0.2, as I recall, right?
Yeah.
Yeah.
Another thing is how do you try to visualize this by microscopical methods to the threshold for perception of gravity?
So do you mean by visualizing sedimentation, for example?
Yeah.
So no, not in these samples. These samples were frozen and went into RNA prep instead of any kind of microscopy.
Yeah, and maybe for the future,
absolutely,
another interesting system for trying to detect and to analyze this secondary pathway would be the use of cell cultures.
Yeah. Right.
Which are very interesting, and could you give the pure cellular mechanism of perception of gravity?
Exactly. I think this distinction you're making is really important, because the fact that we find our statolith-free roots, our starchless roots, are responding at around that 0.2 range, that suggests that that may be the cellular perception, and that the statolith system is imparting greater sensitivity at lower gravity levels. And that may, in fact, be separate. So, thanks.
Excellent.
Yeah. Thanks.
Thank you.
Dr. Wolverton, and we'll have our next speaker. Our next speaker is Dr. Crystal Jaing. She is the group leader for genomics at the Lawrence Livermore National Lab, and she will be giving us a talk on, sorry, Crystal. The microbial tracking in the International Space Station. Dr. Jaing?
Thank you, Kevin. Thank you, Kevin, for the introduction. Very excited to be here. This is my favorite conference that I come every year, the last few years. So as you all know, the International Space Station is a highly specialized built environment. You have heard the term MOBI from Craig yesterday. ISS is a very special MOBI, or the microbiome of the built environment. There's constant dynamic exchanges of the human occupants, the space station environment, and the microbial community, which is composed of various bacteria, viruses, and fungi. So to understand the microbial community in the International Space Station is very important. It is because the microbiome is an important ecosystem that's vital to human and also the environment. So characterizing the microbiome really help us understand, are there any microbes that can cause potential harm to the astronauts? And this also will better inform us to design strategies for mitigation and prevention to really help us for longer term space exploration to the moon and the Mars. So today I'm going to share with you results from a very exciting project funded by Space Biology. The title is ISS, Microbial Observatory of Pathogens, and this is also called Microbial Tracking 2. This is because this is the second series of microbial tracking funded by Space Biology. The first project, Microbial Tracking 1, was led by Venkat from JPL, where he has analyzed or surveyed 8 different locations in the space station. What's different about Microbial Tracking 2 is that in addition to survey the same 8 locations, we also recruited 4 astronauts that we collect samples and analyze samples. The goal is to really help us understand are there any association of the environmental microbiome and the crew microbiome. We have a very experienced research team to carry out the missions for Microbial Tracking 2 study. Venkat needs no introduction. He's well known in the area of space biology and microbiology, and he's leading the effort of microbiology to really look at the viability of the microbes brought back from space station using culture and also targeted assays. Satish Mehta from Johnson Space Center is an expert in virology. He has done some important work to show that herpes viruses can be reactivated in space due to the space environment. This is really important for us to really better understand the potential health impacts So in this project, Satish helped us analyze the herpes viruses from the samples we have collected. David Smith is an expert in aerobiology and space biology, and he oversaw the air sampling and also advised us in better understand the environmental microbiome. Fadi Karua has served as our project scientist since the start of the project, which is about April of 2015, till a couple months ago. He has played a key role in the successful implementation of our project during flight. And so recently, Guoqing Sun has started to serve as our project scientist, and we look forward to working with him for the remainder of the project. So for this project, we have really utilized several state-of-the-art technologies, both to look at the viability and the diversity of the microbes collected from the space station using microbiology and also molecular biology techniques. So our goal is to really have a comprehensive set of data, including taxonomy, gene level analysis, mutation, etc., to have that available as the broader research community. The environmental samples will be deposited into GenLab, and the crew sample data will be archived in the Life Science Data Archive with NASA. So this slide shows the International Space Station surface location we have surveyed. Again, this is the 8 location that Venkat has previously analyzed and published. So these 8 locations, including a port panel next to a cupola, waste and hygiene compartment, advanced resistance, exercise device, dining table, mode 1, overhead 4, permanent multi-purpose module, lab overhead 3, and port crew quarters. Yeah, so for the environmental surface sampling, you can see there's a video playing with one of our crew members doing the sampling. First he put gloves on, and then he go ahead and wipe the surfaces. So we use a polyester wipe, which is roughly about 23 centimeter by 23. And then each astronaut was asked to swipe an area about 1 meter square. So this way we have a really good coverage of the surface area. And so we have 8 surface locations and also 6 air samples. So we have also 5 different sampling events in space station. So overall, we have about 70 samples we have collected in the space station for environmental surface, environmental sampling. Yeah, we also have a large number of crew samples. So total about 352 crew samples we have analyzed and collected. And so this is basically overall a long timeline from pre-flight, in-flight to post-flight. Overall, each astronaut have 8 different sampling points about 6 months before, 3 months before they go into the mission, and 3 sampling points during flight, early, mid, and late. And also 3 time points after they come back, a day after, 30 days, and also 6 months after they come back. And we have basically analyzed several different locations for astronauts, including skin swab, nose swab, mouth swab, ear swab, and also saliva samples. For skin swab, we have 5 different locations in forehead, forearm, axilla, elbow, and navel. So after the samples were collected, the biomass are kind of low. So when we did a sequencing analysis, we actually pulled the samples together for skin analysis. So each of the swabs, we only have 1 sample per time point, except the saliva samples where we have 4 samples at each time point. So this is really to help us minimize the variations between day to day. Yeah, so this is the overall chart to really show how the overall timeline of the 4 astronauts and the samples we have collected. As you can see, we started in about fall of 2016, and we just finished sampling about summer or July of this year. So overall, a very long timeline, but very exciting. We have all this more than 400 samples in our collection that we analyze and get a lot of data. And so each crew, basically we follow them from 6 months before to 6 months after. And then we ask them to collect the environmental samples either in the early or mid phase of their flight mission. So this is shown in the purple square boxes. And so overall, we have the each astronaut do a surface sampling because the crew 4 was able to stay in the space station longer. So we asked him to actually do 2 surface sampling. So that's why overall we have 5 surface sample we have collected total. and also really appreciate space biology and the implementation team to really kind of have the 4 astronauts lined up. We're very excited that we have this continuous sampling and really give us a very nice sample and to the continuation to really allow us to kind of track how the microbiome evolve over their 3 years. Yeah, so for molecular analysis, we have several different ways that we analyze the bacteria, fungal, and viruses. And so this includes a targeted PCR looking for 16S, ITS, and herpes viruses. And we also, this is more targeted analysis to really allow us to do more quantitative analysis of the different bacterial viral species. And we also do kind of unbiased approach. This is using shotgun metagenomics to really allow us to look at everything in the sample. And additionally, we also have used a very unique microarray. This is a large Livermore microbial detection array. This is a really nice orthogonal conformation to really allow us to analyze the viruses, bacteria, and fungi in the sample. So in a single microarray chip, we can analyze more than 12,000 species in the single sample. So today, I'm just going to focus my talk on the data we have collected using metagenomic sequencing. So this metagenomic sequencing, so JPL is the, so they are responsible for receiving the sample and do DNA extraction and then send the samples to a large Livermore lab in my laboratory to do metagenomics and also bioinformatic analysis. And we use the Livermore metagenomic analysis toolkit. This is a very highly scalable Kimber-based approach to do metagenomics to really allow us to look at all the taxonomy level, all the different bacteria, viruses, and fungi present. So this is followed by a number of different statistical analysis and also bioinformatic tools to really look at kind of the diversity, the abundances, and then the distances between different sample types and also between different time points. First, I want to show you the environmental sample microbiome that we have collected. And so I know this is a very busy chart. So basically this is organized, the vertical column shows the 8 different locations that we have sampled. And then the bottom, the x-axis, you can see the 4 flights are listed here, flight 4, 5, 6, and 7. So we started our flight from number 4. This is because of UNCAS MT1 started as 1, 2, and 3. And so you can see there are some similarities. So this is just the top 12 genera that we have, we're showing here just for clarity. And so you can see flight 4, 5, and 7 share a lot of similarities in terms of the most abundant genera we have identified. So the purple bar is Propionibacterium, it's very dominant. And also we see staph as another one really dominant, and also Streptococcus. And one different flight that show kind of different pattern is flight number 6. As you can see, flight number 6, there are several different locations. There's only one dominant genera that's present. As you can see, the first one in the red is the Mycobacterium, which is dominated by that. And again, there's several other locations show a similar pattern, Aspergillus, Methylobacterium, and a few others. And so this is something we actually want to go back to look at the metadata, why flight number 6 is a little bit different from the other flights. And also we look at the top 10 most abundant genera. We also compare that with MT1 just to see how different and how similar the 2 microbial tracking projects are in terms of the top 10 most abundant genera. So the top 3 we have generated from MT2, including Propionibacterium, staph, and also strep. So this top 3 is also among the top 10 in microbial tracking 1. So it does show that there are similarities between the 2 projects, but the other 7 of the top 10 are different. So one potential reason, it could be just a potential reason, is that microbial tracking 2, after sample were collected, we stored that in 4 degrees, and microbial tracking 1, after sample were collected, it was stored in a room temperature. So this could be a reason that there are some differences in terms of the microbial diversity we have identified. Next, we also have a question what locations are microbial diversity more stable, and at what locations they're more variable. So this is an upper diversity analysis we have performed, and shown on the left part of the picture is a child index, which is looking for richness or the number of expected genera present in the different locations. And then the right hand of the graph is the Simpson diversity, which shows the diversity, including both the evenness and abundance of the genera that we have observed. So based on this analysis, we have seen that some areas, including the Crew Quarter and the ARED food platform, which is in yellow and the dark blue, shows they are less stable or they're more variable in terms of overall diversity over the different flights. And so two other locations, WHC, which is the waste and hygiene compartment and dining table, this is shown in the orange color and also pink color. So they seem to be more stable. And so this is really interesting, and we want to really look further in this data in terms of what could contribute to this difference in what we have seen. Is it because there are more exchanges of the human microbiome to the environment, or are there certain species and more dominant that really contributed to this observation? And so next I want to share with you the crew microbiome data we have collected. So as I mentioned earlier, we just completed all data collection, or a sample just got returned back to us in July of this year. So far we have analyzed all the samples from Crew 1 and 2, and we're still in the process of analyzing Crew 3 and 4 samples. So first I want to show you the Crew 1 microbiome from the different body types, different body parts. And as you can see down the vertical columns, this include the ear, mouth, nose, saliva, and skin samples. And then, so this is basically the bottom shows the different days. We have basically pre, during, and post flights. So this is organized by the dates. And so overall, you actually see some similarities between ear and skin, and also mouth and saliva, and nose is kind of distinct on its own. And so for ear and skin, you can actually really see the blue, the green is Propionibacterium acnes as a very dominant. And in ear, we also see staph epidermidis, which is in the dark gray. And the third one is the Malassezia restricta, which is a fungal fungal species that's really dominant in the ear and also the skin. And then the mouth and saliva are very similar, which that does make sense. We do see several different species that are most abundant, including staph, including Rothia. So those are very common that you expect from the oral microbiome. And for the nose, it's dominated by Malassezia, and also Hepatonephilus rhinotitis. This is part of a nose microbiome. So very interesting results. So next I want to really kind of, we have done some analysis to really show, this is really, we start to ask question, are there any associations or similarities of the crew microbiome versus surfaces? So this is just based on Crew 1 data, and also from the first two flight samples, flight 4 and 5. We also included flight 1, 2, and 3 from MT1 just as a comparison. And so this is the data we have submitted to a publication a couple months ago. I just want to share this very exciting data with you. And so basically what you see here are the, so you do see some similarities of some samples. I'm drawing some circles here just to help you kind of see it better. And as you can see, the crew saliva and mouth, which is in the corner there, gray and green colors, they kind of cluster together. And also the crew nose is kind of on its own, the pink color. And then the bottom part, which is the MT1 sample for F1, 2, and 3. So those are the surface samples. They're kind of clustered more in the lower left-hand region. And the ones that clustered most closely together are the crew skin and ear samples, and also the flight 4, 5 samples. And then the flight 4 is more closely related to the skin samples and the ear sample than flight 5. So this makes sense because Crew 1 was the crew that collected F4 environmental samples. So this is really interesting and really kind of really have first indication for us to show the skin samples are really close to the surface samples? Is it because the skin is exposed and constantly shedding cells and shedding microbes to the environment? So that's something that we want to really look further. But the open circle, the open dots on the graph are the control samples. So next, I want to share with you some just recently generated data from a few weeks ago. So for all crew 1, 2, 3, and 4, we actually have analyzed all the saliva samples for all 4 crew members. So just want to share that data with you because it's just very exciting to see all the data together. So basically, these are organized to 4 rows, S1, 2, 3, and 4. These are represent the 4 crew members. And then on the vertical column, these are organized by the time points, pre-flight, during flight, and post-flight. And as you can see, there are similarities, but there are also differences. So this just shows the top 12 genera identified from the saliva microbiome. And just by kind of visually looking at it, just eyeballing it, seems like crew 1 seemed to be the most stable from pre, during, and post. And then as you see, crew 3 and 4 seem to have more variations. And then so we actually went a step further to do some statistical analysis instead of just looking at it. So we basically have done some Kruskal-Wallis one-way ANOVA analysis to really look at which ones are the most differentially abundant when you compare pre, during, and post-flight. And so this is one with a p-value less than 0.05. And so actually we get more genera that are more differentially abundant. Now all of these are on the top 12, which I'm showing here. That's why the number are bigger. So basically for crew 1, there are 57, and crew 2, a little bit less, 10, and then crew 3, 29, and crew 4, we have 56. And so which is really interesting. And so now we just want to take a look crew 4, just because even though it has the same number of the differentially abundant genera, it does look different just by looking at the data on the graph. So this is crew 4. We zoom in just to basically give you a better look to see how different they are, and from pre-flight, in-flight to post-flight. So we also added the most abundant, most differentially abundant ones here in the table. As you can see that the Neisseria, Haemophilus, and Prevotella are very significantly, statistically significant different between the 3 time points, pre-flight, in-flight, and post-flight. So again, this is really interesting and something we want to really investigate further, why these are the most differentially abundant genera and what could cause that difference. So next, we also look at crew 3 because they also look really, really different. And so crew 3, only one of the top 12 shows a statistically significant difference between Neisseria. This is shown in the orange bar here. You can see in pre-flight, the orange bar is much larger. And also post-flight, it seems to be back to normal. But in flight, that bar has really shrunk a lot. And this is kind of sort of being more dominant by Streptococcus, which is the yellow region. Another really interesting fact about crew 3, he's also part of a study of JAXA study to really study the impact of probiotics. So I included the title of the project here, a study for evaluation of impact of continuous consumption of probiotics on immune function, intestinal microbiota in astronauts and a closed microgravity environment. And the prebiotics that he has taken is Lactobacillus casei strain and Shirota. And so that's a really interesting fact we want to follow up. What does the probiotics have done to the crew 3 microbiome? So this is additional analysis. Actually, in addition to the genera level, we also look at the species level. Does the same trend follow in terms of the microbiome differences, pre-flight, in-flight, and post-flight? We actually see a very similar trend at the species level. And we do see 3 species, overall about 128 species that are differentially abundant, pre-flight, in-flight, and post-flight. And among the 128, 3 of them are also among the most abundant, most abundant in the top 12 that's shown here in this graph. And so we do see these three, Neisseria, flavescens, Fucobacterium, and Porphyromonas. And so these three are all part of the oral cavity. And so again, it's very interesting that these are the three that most differentially abundant pre-in-flight and post-flight. And additional analysis we have done is to look at offer diversity. So we want to really see, are there any kind of changes in overall richness or the number of genera from pre-flight, during flight, and post-flight. So this is a violin plot to really show the distribution of the offer diversity of the 4 saliva samples at each time point. And as you can see, there is a decrease in terms of the richness of the genera in saliva. And also they seem to rebound back to normal in post-flight. So there are a number of questions about group 3 we want to follow up on. And so for example, so these are the questions we want to ask. Do probiotics decrease overall microbiome diversity? And do they have an influence on the abundance of certain microbes? And we would like to really kind of compare, if we can talk to the JAXA collaborators, is something we want to do for the study, because they did collect gut microbiome data. We really it'll be nice to really compare the gut versus saliva microbiome. Additionally, as part of their study, they have collected immune biomarker data from salivas. So it'll be really interesting to really look at any kind of connections or associations to do any predictions in terms of immune microbiome, immune biomarker to microbiome, pre, during, and post-flight. So now we want to put all the data together. So this is everything we have analyzed from crew members. So we have 4 crew members we have analyzed, and we have analyzed the crew 1 and 2, the complete data set. For crew 3 and 4, we have only analyzed the saliva and mouth swabs. And so it's really interesting that you can see there are some patterns you can see here. So this is basically done by T-SNE analysis. This is the T-distributed stochastic neighbor embedding technique. It's a very powerful technique to really reduce the dimensionality to something you can visualize. So this is basically a comparison of both diversity and abundance of the microbial species that we have analyzed. And so if I can draw some circles in terms of the sample types, the crew, saliva, and mouth samples kind of cluster in one area. So you can see crew 1, 2, 3, and 4. This just shows that even though they're all saliva sample, but they are different crew member to crew member. So they carry their own distinct microbiome, but it's different from surface mouth samples and other surface and ear and nose swabs. So the surface samples are kind of clustered together on their own. So these are all the surface samples from our study, flight 4, 5, 6, 7, and 8. And also the crew skin, ear, and nose samples kind of cluster on its own as well. Again, this is really kind of interesting to kind of validate our early analysis on crew 1 sample only, which we have seen a close connection of the skin sample to the flight 4 and 5 sample that he, especially crew 4 sample, the flight 4 sample that he has collected. And so the pink color there is the skin sample. So this is really kind of a very interesting, something we really want to follow up on, the skin sample that's mostly close related to the surface samples. And my colleague, Mike Morrison, will really talk more in terms of our observations and our analysis in bacterial exchange between astronauts and International Space Station. So welcome you to attend his talk on this Saturday. So we still have a lot of analysis are underway, just because we have collected a lot of data. It's very exciting to really mine the data using additional bioinformatics, statistical analysis, really better understand some of the questions are listed here. So we have done a taxonomy analysis. So our next level analysis is really to do gene level analysis, looking at antibiotic resistant genes, for example, virulence genes, metabolic genes, to look at the changes pre, during, and post-flight. We also really want to see kind of similarities and differences and the more kind of strain and genomic level. So far, we've done the genera level and species level, but we really want to go a step further to really look at strains that are in mutations. We can better track and understand the microbial exchanges between crew members and the environment. And so we also really want to look at kind of the space microbiome to Earth microbiome by looking at both the crew members and also the surface microbiome. And also very importantly, to understand any potential implications of the certain microbiome changes to crew health. So finally, I really want to thank our collaborative team, so that we have a fantastic team, very collaborative, and really work together really well. And so this is a picture from a couple years ago at the Seattle ASGSR. I really want to thank the team for the great work. Also want to thank Space Biology for giving me this fantastic opportunity. This is my first space biology project. I'm very exciting. And I really want to thank the Space Biology program to really give us this opportunity, and really are very grateful. Thank you.
Nice talk. Can you tell us anything about any of the illnesses the 4 astronauts had during the course of the flight to associate with their microbiomes?
Currently, I'm not aware of any illnesses, but that we are still waiting to collect that data. So we had a questionnaire generated for each of the astronauts. So we are in the process of compiling that information. We don't have that information yet, but it's something on our to-do list. It will be really important to get all the metadata together when we start getting working on our report and publications.
So I noticed that you had some saliva samples, and I'm curious if you correlated those at all with the presence of cavities or other dental health aspects of the astronauts. Is anyone looking at that?
That's a really good question. Again, we had a questionnaire in terms of, I don't believe we checked their dental record. So that's a really interesting question. We can certainly go back to Swiss biology and maybe HRP. David, I'm looking for your input. And if that's something we can obtain, I think that will be really interesting to put together. Yes.
As usual, I have the same question as Keith. The reason is you saw the elevated levels of Streptococcus and Haemophilus in saliva. Otherwise, thank you for answering Keith's question.
Okay, thank you.
Hi, so just kind of pulling off of their question. So are you going to at any point start looking at activity of these organisms, even if it's transcriptional activity, to see they may be not doing anything in that environment?
Yeah.
And the second part of that is, now that you've seen probiotics can really shift the diversity of the community, are you going to start doing more correlations with the diet of the astronauts and really see how the food supply is changing your community?
Yeah, that's a really good, excellent question. So first question regarding the activity. Our team has discussed one time if we should look at transcriptome of saliva samples. I think that's something still on the agenda, and we will have more discussions on that. And then, so OneCat has collected viability samples. So we do have culture isolates from the sample we collected from the space station. So that's something we could do further analysis on to look at transcriptomics of those samples. And your other question related to diet, so that again is a very important question. So we would like to really kind of explore if we can get their diet information, part of the questionnaire, and maybe some additional metadata we need to obtain. So that I think that would be really interesting to put a story together. Thank you for the comments. Yeah, great, thank you.
Thank you, Dr. Jaing. And our final speaker of the morning is Dr. Rémi Canton from the French Space Agency. He is the project manager for Declic, and he'll be speaking about Declic physics in the main laboratory ISS research studies. Dr. Canton.
Good morning, everybody. So thanks for the introduction, Kevin, even though I'm not a doctor, I'm just an engineer. Nobody's perfect. So I'm in charge of microgravity projects related to human spaceflight at CNES, the French Space Agency, in case you haven't noticed my accent. So I won't present you any scientific results. It's going to be more like programmatic and give a status on the Declic project. So Declic, it's either a French or English American acronym. It stands for Device for the Study of Critical Liquids and Crystallization. And it's a joint NASA CNES program in physical material science. And it studied two main things, critical fluids and directional solidification and characterizations of those fluids. It's currently housed in Express Rack 4. It's made of two SSLs, single storage lockers. One is mostly like the optical part with the laser cameras and everything. And the other is electronic, is housing thermal and power data handling. And so that's what you have in the red dotted line. And inside the Declic facility, you can put an insert. It's like a big shoebox if you want. That's where the science is made. And it contains the scientific cell. So Declic was launched 10 years ago and been operated for like 10 years. We'll give you a status. And it's operated from CNES in Toulouse. So I'll show you. So for those of you who don't know Declic, you actually have already seen it if you watch the Martian movie. So I'll show you. So that's me operating Declic on a bad day. And on a very bad day. So Declic is ahead of us. So I will explain why they use. It's actually the real model that you can see on the screen. I will explain why Hollywood is ahead of us. So like I said, we have different inserts. We currently have three. One is called Ali. It studies critical fluid, and it's sulfur hexafluoride near the critical point. Another one, sort of similar, HTI for high temperature insert. It studies water around the critical point. And the last one is a DSI, directional solidification insert. It studies the solidification for transparent alloys. Of course, we would like to see it in metals, but metals are not transparent. So we use a succinylnitrile to observe the solidification. And in the room, you have all the PIs involved in this team. So how Declic is organized? At CNES, we are the payload developers, and we are also in charge of operations. And it involves 16 American and French laboratories, and they mainly around the inserts, because that's where science is made, and they provide support for operations as well. And throughout the years, because it's been 10 years, there have been numerous major publications with strong impact factor. NASA also has a big role because we wouldn't be in the station without them. So NASA is in charge of transportation up and down of the Declic facility and of the inserts. You're going to see the different cycles. Express Rack 4, like I said, so they provide us power, water, communication, and crew time. It's an automated payload, but since we have three inserts and we only use one at a time, you need to change them from time to time, as well as hard drives. So and crew time is a precious resource, so NASA allows us to use some of it. So this is our control room in Toulouse, France. And we perform real-time operations, and we connect through POIC, Payload Operation and Integration Center, down in Huntsville, Alabama, at Marshall. And we connect also in real time with the scientific teams, with the PIs, and they help us they guide us through the operations. So from the beginning, we've done 839 days of operation. So if you follow, you're like, well, 10 years is more than 800 days. It's because we do 3 weeks of operations, then 3, 4, 5 weeks without, because we cannot have like a scientific team every single day on the phone, like 24/7. And also there have been different interruptions of the mission. I will explain a bit in a few slides. So it's been spread over almost 50 flight sequences. So from increment number 21, 22 up to 57, 58. And it generated a lot of data to keep our scientific team busy. So I have a slide for each insert. For the ally insert, we had the 3 initial scientific objectives. One was study the boiling phenomenon, and in particular the triple line behavior. Study also the turbidity of SF6 near the critical point, and the liquid gas phase separation has been studied as well. So that was the science objective for ALI. And we returned it to the ground early 2014 to change the cell, to replace it with a still SF6 with a density as close as possible from the critical density. Returned to the station 3 years later. It doesn't take 3 years to replace the cell, but during that time, other inserts were operating. And LIR, R stands for reflight, or refurbished, if you will, ran basically throughout the year 2018. And the data is still being under analysis. For the directional solidification insert, same thing. We had the first run up to June 2011, and studying like the dendrites, like during the solidification, and the instabilities and multiplates and oscillating patterns. We changed the cartridge when we returned the insert to the ground to change the camphor concentration. And we had a second run, DSIR, through September 2017 to November 2018. And the idea, so it's still about the station. The idea is to change to bring it down to earth again, to change the cartridge and do it with another concentration of camphor. The last one, HTI, like I said, so it's studying the water around critical point. We study the critical temperature to determine the critical temperature as precisely as possible. So we had a 1 milli-K resolution, which is pretty good. We demonstrated the ability to tune a radiothermal gradient inside the insert, and that's a preliminary step towards saltwater studies, because initially it was pure water. So when we returned to the ground in 2011, we changed the pure water cell with a saltwater mixture called SCWM, and operations resumed between 2013 and 2017. So the program has been completed, and that was an important step towards the next one we want to do, is to the study of oxidative processes. I also returned HCR in September 2 years ago, and we are currently in the refurbishment process. And depending on the results of HCR, the scientific team in Bordeaux will decide if we do another binary mixture, like water and salt, but at different concentration, if we do a ternary mixture, water, salt, and CO2, or another binary mixture, water plus another organic compound. And the idea is to have it returned to the station in 2021. So, Declic is not closed, but for refurbishment, because 2 years ago we had a degradation of the laser. That we have the ground model on the screen, and in red that's the laser. We had the voltage and current really decreased. Pretty much the laser died, and despite camera setting during that phase to increase the contrast and the gain, the interferometric images were not doable, exploitable. So out of the 3 inserts, 2 were affected, DSI and LA, because it needed interferometry and scattering. The bad thing is that the only 2 on board the station were DSI and LA at that point. HTI was on the ground, so we could have run HTI, except we didn't have it. So we did an exercise with different PIs to see, okay, without the laser and without interferometry, what can we do? Is it worthwhile pursuing some science, or do we bring everything back to Earth? So it's been decided that these interruptions we could allow to do degraded science. So we've been able to do degraded science, but interesting science still, for like a year until the end of last year. And in parallel, the idea, so even though we were close to finish the initial scientific program, everybody from NASA and CNES point of view, either programmatic or scientific community, wanted to extend the program up to 2024 or beyond if the station keeps going. So we needed to secure the current program. So that goes with repairing and upgrading, if possible, the current facility, and to give an extended lifetime. And once the Declic scientific program would be over, to have another program called Declic Evo, like evolution. So the evolution part is not really in Declic, but in the inserts. That's why earlier this year, in January, we returned the Declic facility to the ground on SpaceX 16, and is under refurbishment and upgrade. It will take another year and a half, and to bring it back to the station early 2021. So to give a status of what we've done since January, so what we call phase B is like the analysis part, because okay, we know that the laser needs to be changed. Well, first we need to find a new one. Obviously, something that has been designed 15 years ago. You have like a big obsolescence analysis to do. The laser manufacturer doesn't exist anymore, so the laser we had to change to find another one with the same physical properties. If you remember the picture, it's like a shoebox with very small room. So to make sure that the optical path is not changed, that you have the same optical properties and everything, it was really tricky to find actually to select the laser. So it's about to finish its performance and environment test, and we already selected the flight model. That's very good news. And for all, and obviously since we have it on Earth, we don't want to change only the laser. We do an analysis to see what are the life limited elements, what needs to be changed. We don't want to. It's a bit of a frustrating exercise, because from an engineering point of view, we would like to change pretty much everything. But if you do that, it's a completely another scale of project. We wouldn't be able to do it in time. So we need really to pick really what needs to be changed to make sure that the extended lifetime with the level of risk is acceptable before returning it to the station. So this analysis phase will be ending before Christmas, and 2020 will be dedicated to the actual change of the hardware. And regarding the inserts, at last year's ASGSR, we agreed with NASA on 2 new inserts with their respective scientific objective, with Frank Monte, who's over there. And we, during this year, since last year, we did phase A, like a feasibility analysis, to try to convert those scientific objectives into an insert. And again, which is constrained because it has to fit within Declic. So not just in terms of size, physical size, but also interface and performance. We have the cameras we have the data downlink we have. So we have to constrain the scientific team, which are very creative, but we have to be in this framework. Back. Wrong button, I guess. All right, so the next insert is, one of the new inserts will be called SCORE, for supercritical water oxidation. It's the logic follow-up of HTI insert. Like I said earlier, we had like the pure water cells, then mixed with salt, and the next step is to see how to study oxidation and to make organic combustion. Water at the critical point is a perfect solvent, so it can dissolve organic matter. And what's interesting is it doesn't, you don't have any off-gassing of NOx, of nitrogen oxides. So that's very interesting. You have CO2, but CO2 we know how to get it. But avoiding NOx is very important, and it's a priority for exploration in NASA, for NASA Vision and CNES as well. And that's why you had Matt Damon using it ahead of what we're already doing, because it will be a perfect way to burn a waste in a clean combustion way. So for that, we'll work. The study for the insert is to use mini fluidics to, instead of running batches like we do on HTI, to have a continuous combustion and changing the ratio. The second insert is called aerosol. It's to basically the idea is to create small cloud and to study the droplets. The advantage of creating a cloud, you do it by doing a adiabatic expansion, is you avoid, in microgravity, you avoid the boundary effects. The cloud is floating and not touching around it, and the droplets are not falling. And that's why we want to study the interaction of droplets, like coalescence, and also in a turbulent mode. We're going to create turbulence, and to see how the different interactions of the droplets, how the energy is transferred. And that's very important also for, I mean, the reason why we want to do that for science, it's very important for to improve weather models. If you look at all the GIEC reports, one missing cornerstone of the weather models is to really understand better how it works within the clouds. So I think that that's a small that's creating the small cloud, but on the ground, so you see the droplets, but they are falling. So we like microgravity to avoid this rain. And the last insert is not really a new one. If you remember the DSI slide, we need to change the cartridge another time, and what is missing from it's like a lesson learned from the previous insert, the previous version of the insert, is having a really precise temperature measurement at the liquid-solid interface. So that's what will be added on top of the different concentration of camphor. In terms of schedule, so we are at the red line. So as you see, like if you look at the blue bar that's on the left, SM Declic, that's when we operated Declic. It's now on the ground. We want to bring it back up in 2021. In the meantime, so we refurbish it, and upgrade it, as well as the HTR insert. So when Declic is back up, we're going to run the Ali and DSI inserts because they remain in the station. So Ali is supposed to go first because he was supposed to go first. And then DSI, that might change because DSI, the succinylnitrile is, with time, it gets darker. And since it needs to be transparent, if we can't review fold weighting more, we might invert LI and DSI. Then we run HTI R2, the one that's on the ground. And for DSI R2 to run, DSI R needs to be back down. So that's why maybe inverting Ali and DSI will be interesting for that point of view. But obviously, if you ask the scientists for Ali, they want to run first. That can make sense. And we have the two inserts. You have the gap. I will explain a little bit. We have the two new inserts I told you about a bit later. Well, first we need time to make them. And the room we have, it's a bit programmatic. There are still discussions to add another insert, probably the follow-up of the Ali insert. So we currently have discussions offline during the ASGSR to see what we can do during this time. It can be for margin or hopefully for more science. So 2024 is the theoretical end date, but the life expectancy of the Declic Evo is way beyond that. And I'm sure the scientific team will manage to keep us busy beyond that date. So that's why Declic is on its way to be refurbished into Declic Evo and with an extended scientific program. So thank you very much for your attention.
Questions? Anyone?
So nice presentation. On the SQUO, the supercritical water oxidation, can you elaborate on that one a little bit about the application for exploration?
Well, yeah, a little bit. During transportation in a spacecraft, and once you land somewhere, I mean, you obviously generate a lot of waste. The trend is to try to close the loop between, instead of generating waste and not using them, it's trying to recycle as much and to make something out of the waste, and at the same time not polluting. Like I said, with the NOx, if you, the easiest way usually to get rid of waste is to burn them. And you can, I mean, inside spacecraft, you can't really do that because of all the off-gassing that you have. So that would be very interesting for long duration transportation. And also once on Mars, you know, like Matt Damon Lake, that's what we want to take advantage of the properties of water around the critical point to our advantage in terms of, when I say pollution, is really, I mean, it's not in a philosophical way, but in a very practical way.
very much.
Thank you. Well, if you have questions on science, I can denounce the PIs in the room.
All right. Well, thank you, everyone. And I'd like to thank everyone for this morning's session. It was very informative. And now we're off to, there's a break. And then afterwards, please go to all the different technical sessions and remember the 2 programmatic sessions this afternoon. And also, I just want to remind everyone, tomorrow, Friday, is when we begin the undergraduate and graduate student poster session, and then Saturday will be the high school. So please attend those starting tomorrow as well. Thank you, everyone.